talk in this nanomotor series. I was not aware that it existed before, so now I will carefully. So yes, I'm a researcher at the University of Bordeaux in the LOMA, which is Laboratory of Wave and Matter. And I'm going to present you this work that I actually did when I was at the University of California, San Diego, in the team of Jeremy Palachi. And this is the work we did in collaboration with Quentin Martinet. And so I hope you will enjoy it. So to begin with, so I'm not going to do all the introduction about what is an active particle and why it's maybe important to have to study nanomotors, because I guess that you're already very familiar with all of that. So I'm just going to tell you directly what we wanted to do, and what we're actually interested into was to see how we can control the assembly of active particles to build specific kind of machines or devices. And actually, so they've been used in different ways and different ways have been used to control their motion. So the first one is using, for instance, geometrical boundaries, which is what you can see here, and you're probably aware of this work already, where you have genus particles that get cornered into a gear and make it rotate, which is their persistent motion. And here in this case, so it's a work from the team of Roberto Di Leonardo, where they built microgears using laser lithography, and they immersed it in a bacterial bath, and the bacteria come, and by harnessing the motion of the bacteria, they can transfer their momentum to make it rotate. They could build like hundreds of these gears. And another way is to control the motion of active particles and harness them is to use, for instance, light templates, which is what you can see here in those examples, where you have a bath of bacteria that are light sensitive. And the author, what they did in both these studies is that they made the bacteria light sensitive, so that their propulsion speed depends on the light intensity. So that when you have a light intensity, the bacteria will go faster or slower depending on how you build them, you modify them. And therefore, you can tune the local density of bacteria by tuning the local speed that you impose your bacteria and build those kind of patterns. So you have all these great works that show that you can actually control the motion of the bacteria to generate work or to build some kind of self-organized pattern. But what we were interested is to see if we can now use active particles and assemble them in specific machines. And as I'm going to show you in this talk, what we can do is use optical templates, again, to assemble these micro machines from smaller colloidal machines, which are the active particles. So the main building blocks that we used before, I'm showing it to you here. It's just a micrometric particle with a photocatalytic part at the back and a polymeric part at the front. And this particle, when you put it in a solution of hydrogen peroxide and you shine light on it, it decomposes the hydrogen peroxide, which triggers its self-propulsion by diffusiophoresis. Okay, and what we need to remember is that just the velocity, typical velocity that we would get, is proportional to the local gradient that is generated. And these particles, so if we just shine a uniform blue light in the sample, the sediment at the bottom surface of the sample, so here everything is in 2D, and they exhibit a persistent random walk, where they go in straight line, and then thermal diffusion orient them, and they show this effective behavior at longer time. So we have this self-propelling and asymmetric machine, and the particularity of this heterodimer is that it's composed in two parts, and actually it's designed this way so that it can be sensitive to light gradient. And this is we actually harness before this particularity to direct the particles and force them to self-assemble. And this is what you can see here. So this is part of our previous work. Well, if you just put a single laser spot, like even a crappy one, you see that it's kind of ugly here, the particle will reorient all the time towards the laser spot. And using this particularity, you can force the particle to come together, interact, collide, and at some point you will see that they can form some kind of structure like this one, which is a spinning microgear. So this is something that we saw before, and we could build many of these microgears, and we studied this behavior. But the main point here is that the way they assembled was through self-assembly. And if you use self-assembly, what you have is trial and error. Success every time. At some point you may success, but it's not always super robust. And also, like the kind of structures that you can make is limited. So you don't have specificity over the kind of structures that you want to build. The question is now, okay, how can you go beyond this and build a specific structure? So if you have your bricks, a way to do that is to find a way to assemble them in what you want. And at the microscopic scale, if you have a colloidal particle, what you can do is you use your colloid and use what we call optical tweezers to organize them or assemble them the way you want. Okay? So what we're interested was to see if we can use optical tweezers with active particles to assemble them the way we want and then create dynamical superstructure. So we used very briefly this optical setup that I'm showing you here, where we have the sample here. And we have, can you see the mouse actually? Yeah. Okay, I'm gonna assume that you can see my mouse. So we have the sample and we have one blue light that uniformly illuminate the sample and one red light that is directed onto a spatial light modulator, which is the SLM here on the left. And that allows you to design whatever type of pattern you want and send it a pattern of light intensity that you want and send it on your sample. So a spatial light modulator basically is just an assembly of a small mirror for each mirror you can adjust the phase of your incoming beam. So that what happens is that instead of having just one beam, at the end you have multiple beams with different phases, and you can build a phase pattern so that all your beams will interfere together to give you a desired specific intensity pattern. That's how it works. Okay, so that's for the experimental detail. And now just a quick reminder about optical forces, just to make sure that we are on the same page. So what do they come from? So they typically come from the transfer of momentum of incoming photons to particles. So that if you have an incoming beam and a particle which is immersed in the medium with a refractive index higher than the medium, if you have more photons coming on one side than on the other side, then you will have more photons refracting in one direction than in the other direction, which results in gradient forces. It will pull the particle at the center. And at the same time, you also have photons that are coming, that are reflected back, or that can be absorbed, which will push your particle forward. Because then by momentum conservation, you have, if the photons are going in the reverse direction, your particle has to be pushed forward. So if you have just a dielectric particle, usually you can trap it stably at the center. But now what happens if we have our heterodimer particle, so the one that you can see at the top right of the slide? In that case, you add the hematite part, which is a metallic part and which is strongly scattering. And since it's strongly scattering, if your particle is not swimming and you're just trying to trap it, then you can't trap it. Basically, you will just push it away because the scattering force is too strong. So the radiation pressure is too strong. You cannot trap it when your heterodimer is at equilibrium and when it's not swimming. So now what happens when all your particles are swimming? Well, in that case, this is what you can see here, where you have a laser spot. Your particles are all swimming, and you can see that you have your particles that get trapped in the center of the laser spot. And then if you release the trap, they just go away, and you can wait for another particle to cross the laser spot to be trapped. Okay, so we can now compute the lifetime of the particle in the traps. This is what you can see here on this graph. And you can see that for an incoming power of about like 0.5 milliwatt, you have a very stable trapping with a time of more than 100 seconds. And what you can see is that for this type of power, you have a trapping force, so lateral trapping force of typically 0.1 to 0.2 piconewton, which compares to the propulsive force of the swimmer. So what it tells us here is that we have an interplay between self-propulsion and the optical forces that enable the optical trapping. So from where does it originate? So we have two main competing phenomena that enter into account to understand what's happening. So you have a particle that's coming from the left, and as it's crossing the laser spot, you have two torques that are induced. First, the one that is due to the gravity, to the fact that the hematite is heavier than the plastic part at the front, and which is due also to the fact that the swimmer is swimming close to the surface, interacting with the surface, generating hydrodynamic flows that are reflected on the surface, and also chemical gradients that interact with the surface and modify the orientation of the heterodimer near the surface. And you also have an optical torque that acts on the particle when it crosses the laser spot to orient it against the direction of light propagation. So the first one, we can actually estimate it from the experiment, and how we do that is by just analyzing the mean orientation of the swimmers, which are also submitted to thermal fluctuation in the absence of any beam. So you remove the light beam, the red beam, and you just look at the mean orientation of your swimmer. You can get the distribution of the orientation. And from the distribution of the orientation, it allows you to extract and estimate the torque that is induced by the interaction of the swimmer with the surface and all the other effects that happen basically in the absence of your optical beam. And the second one, the optical torque, can actually calculate it and compute it for a heterodimer by considering the shape of our particle. We can do that. This is what I'm showing you here, where you have the force along the x direction and the force along the z direction on the right for both the polymeric part and the hematite part here in black. So for the polymeric part, you see that the main force the force that is the dominant, at least, the force along the x direction, so parallel to the surface. So basically, you have a gradient force that tends to keep the particle at the center of the shot. And for the hematite part, in contrast, what is dominant is a scattering force that will tend to push the hematite parts upwards, so to eject the heterodimer. So we have these two forces that we can estimate, and now we can summarize that in this graph that I'm showing you here, where you have the two torques as a function of the angle of the heterodimer when it's at the center of a laser beam. So the first one, so the one that we extracted experimentally, which is due to the interaction with the surface here at the top, it's a blue circle. And the other one, you can see that for values of typically between 0.2 and 0.7 milliwatt, we have values of the torques that we compute that compare with the one induced by the surface. And of course, the optical torque increases when you increase the optical power. So we have this range of power for which we have a comparable value of the two torques. Okay? So we can compute that, and now we can analyze in more detail what the dynamics will be, and we can distinguish three situations. The first one is when the particle is crossing the laser spot. So it's basically the trapping force is not enough, so it's just crossing the beam like if it was not here. And you have the second case when the particle is ejected, so it's pushed upward. And you have the last case which is the one of interest, it's when it's trapped, it remains trapped at the center. And we can summarize that in a phase diagram that you can see here, where you have the velocity of the swimmer in the phase diagram that comprises the velocity of the swimmer and the incoming power beam. You can see that when the power is low, the particle crosses the trap. When it's too high, basically, the particle is ejected. And you have this range of values for which you can have a stable trapping at the center. And what we can see is that by looking at the phase diagram, that we have an area which corresponds very nicely with our experimental value. What it means here is that really you have a trapping that is completely enabled by the activity of your particle. You cannot trap them in the absence of activity. So now we can use one optical trap to trap one particle, but you can use also multiple trap to trap multiple particles using the holographic tweezer setup that I presented you before. This is what you can see here, where you initially have all the particles swimming randomly in the sample, and then you switch, you put an optical template, and you wait for the particle to fill up the traps. And that's what you can see, and you just have to wait, and you see that they form this pattern here of individual traps, where you can write UCSD. Okay? And so the nice thing here is that you have a fast filling of the optical template where you don't have to move your optical traps. So you don't have to use a pick and place strategy. since your streamers are effectively diffusing very fast, you can fill the traps very fast as well, which would take like 100, much longer, at least if you were just waiting for the particles to fill the trap when they are at equilibrium. Okay, so we can organize the particles the way we want, but what interested us in the first time was to see if we can build some specific colloidal architecture. And we can actually do that. And the idea is now, instead of using individual traps that are spaced, you use a closed packed arrangement of optical traps. So the idea is that you impose a template of traps, you wait for the particle to fill the traps, to build a specific architecture. And once the architecture is built, you just release the optical traps and you see if you can get some kind of stable structures through it. And we could demonstrate that for a wide variety of micro machines. So basically more than 40 different micro machines that we designed a priori. And you can play with the structure to get some kind of rotating machine, carrier machines, or some kind of other assembled machines that would exhibit more something like a translational behavior. Okay, so how can you understand all this behavior now? So what we can see is that if you look at the structure here that I schematized at the top, you have a different swimmer orientation depending on their location inside the structure. So the individual swimmer's orientation depends on the local geometry. And actually, you can distinguish different situations. So when you have one swimmer trapped, it's say vertical, when it's free, it's alone, and when we have a swimmer with only one or two neighbors, the structure is unstable and your swimmer will detach. However, when now you have 3 or 4 neighbors around the swimmer, in that case, it's stable, the position at least is stable, and your swimmer will have a well-defined angle in average with respect to the local tangent. For 3 swimmers, you have typically a pi/4 angle that is formed with the local tangent, which is what you can see also on the scheme. And for 4 neighbor swimmers, you have something which is more like a pi/2 angle, so basically an orthogonal direction with the local tangent. Okay, and what happens here is that all your swimmers actually, at the boundary, they constitute an active boundary, so they are pushing your structure to generate a propulsive force. And now you have the last case when you have 5 or 6 neighbors around your swimmer. And in that case, you have a geometrical frustration. And your swimmer that is surrounded by 5 or 6 neighbors stays vertical all the time. And since it's staying vertical, it generates an attractive pumping flow because it wants to swim. But basically, that's the bottom surface that prevents him to swim. So it's pumping the fluid. So the situation is exactly the same as what you would have if you're in a small boat and you're trying to paddle against a wall. You will just like pull the fluid behind you. So here it's the same. You have a swimmer that's trying to paddle against the surface. So it's just pulling the fluid behind it. And it's contributing this way to attracting all the neighboring swimmers, and it's contributing to the cohesion of the overall structure. So what we have in the end is a propulsive boundary here and a cohesive core. Okay? And actually, from that, we can understand and predict the dynamic of the structure by just considering these 2 ingredients. Where in the first case, you have you have only the active boundaries that contribute to the rotation or to the translation of the structure. So you build this very simple equation. And what you can see is that you can, by considering the average orientation of the swimmers at the boundary, you can predict the rotation rate of the structure. You can do the same for the translation. And this is what I'm showing you here. Well, what you have is here the predicted speed as a function of the actual measured speed. You see that you have a very good correlation between the two by just measuring the instantaneous orientation of the swimmers. And you can do the same for the translational speed, where you measure all the orientation of the swimmers. From that, you can estimate the average translational speed. What it means here is that you have a dynamics of your structure that is completely encoded in the shape and the arrangement of the individual swimmers inside the structure. Okay. And so one nice thing also is that your swimmers, they have a defined position, but they have actually the ability to switch direction or the other. So one swimmer that stays horizontal and contribute to the active boundary can switch to a configuration where it knows now contributes to be part of the cohesive core, provided that you can modify the local environment. So that's why we call this the title of the paper, Metamachine made of a pluripotent colloid. The pluripotency is this ability to switch between one configuration between 3 swimmers to one configuration between 4 swimmers if you want, or 5 swimmers and 6 swimmers. And the swimmers can reprogram between one or the other configuration. And this is what so now that is for the explanation. The active particle can switch functionality, and you can actually use this particularity to reshape dynamically different structure, like I'm showing you here on this movie, where you have a full process, where we started by building 3 rotors with 4 individual swimmers that are trapped. And you can see the optical template that we used at the bottom right of the movie here. So we start by building this structure. So you have each swimmer inside your individual rotors. And then what you do is you merge everything to change the individual configuration of your swimmers. So you have each swimmers that became core swimmers, individual swimmers that became part of the bigger structure. And now you can remove the edge swimmers to get a smaller structure and so on. You can diminish the size again, so that you have core swimmers that become edge swimmers until you reach the smallest configuration possible, which is this one. And in that case, we just trap the center of the structure. And what is important to remember here is that the motion that you just saw is not forced by the optical tweezer. It spontaneously emerged as a result of the shape of the structure. So in all those movies that I showed you and that I will also show you after, you have uniform blue light activation, and we use the template just to build the red template, sorry, just to build the architecture, and then we remove it, and the architecture is autonomous. So we can reshape that, and now I just want to show you a different example to more illustrate a bit more of how we can program the functionality of our structures by playing with its geometry and shape. Here you have 2 different configurations. Well, we start by building these 3 ellipses here, and you see that the arrangement is slightly different. And actually, we play on this just slight difference in arrangement to get 2 different rotating structures that rotate in opposite direction. And here, this rotation comes from the fact that you have actually more swimmers that are pushing on one side of the helix than on the other side, which makes it rotate. By just playing with the shape of your structure, you can get a very persistent dynamics with a rotating structure. So we can build rotating machines, but we can also build something that exhibits more like a translational behavior, which is what you can see here. We have 4 individual swimmers, 1 central wheel, and 2 ellipses. The same thing, we start by building them, and then we merge the overall structure. And what you see is that we pause it, you have this structure here with a frustrated orientation of the swimmer, which is at the center and inside the cavity here, which results in more swimmers pushing at the back, so constituting the active boundary at the back than at the front. And in turn, what you have is something like a translational behavior because you created a forward-backward asymmetry, so kind of a micro rotor, but at a larger scale, which presents this fore and aft asymmetry. And actually, so I'm showing one example, but with different kind of a translator like that, having this an identical forward asymmetry. Okay, so I think I've showed you almost everything that I wanted to. So this brings me to my conclusion. So you could see, I hope that I've been able to show you at least that we can use the interplay between optical forces and activity to build autonomous and reconfigurable micro machines, and that hopefully it will help to open new roads for the programming of the various kind of active objects. We are also still studying in the lab of Jeremy Palachi. And here you can see it, the full list of the meta machines that we could create. And with that, I would like to thank you for your attention, and I would be happy to take any questions that you would have.